is a great pleasure for me to introduce Bob Cook. Bob Cook received his PhD at Berkeley with Al Riley, the same lab that I received my degree at, but mine was a few years before Bob's. Although I've known Bob for about 30 years now, and we see each other at conferences and talk a lot about research. Bob went on to do a postdoc at the Houston Health Science Center with Tony Wright, and then went on to Tufts University, where he's currently professor and department chair. Sorry.
Me too.
Bob is one of those scientists who became enamored with computer technology, but unlike many who love computer technology, Bob never lost sight of the goal, which is to understand the behavior of animals. And he used that technology in extremely innovative ways to study things that people don't typically study with animals. Texture perception was one of the areas in which he made major inroads and major advances. Same different concept learning. We thought we were very clever at looking at same different learning with shapes and colors, and Bob took more naturalistic stimuli like birds and flowers and showed that you could get much better transfer effects than we ever got with colors and shapes. Equivalence class formation, we did some work with simple colors and shapes. Bob with much more complex stimuli. Object perception, picture perception, motion perception. Bob made remarkable advances in those areas and did research that very few other people were engaged in. More recently, Bob engaged in a project to ask what the memory capacity of pigeons might be, and discovered that they can remember literally thousands of different slides and perform above chance with arbitrary associations of a huge set of S plus stimuli, a huge set of S minus stimuli, and showed that they could remember these stimuli at significant levels. Recently, Bob has taken on, in addition to his administrative duties, he's co-editor of the Comparative Cognition and Behavioral Review, electronic journal, which is innovative in its own right, but also an excellent journal with Ron Weissman. Today, Bob is going to talk to us about stimulus control and the way that it relates to this exciting area of research that he's been engaged in.
Thanks. You know, Tom, I still remember it was in San Diego at Psychonomics. You were like the first. It was like the first Psychonomics I ever went to, and you were the first person I ever got to sit down and talk to for an extended length of time. You were very polite to listen to me for a very long time, talking to a young graduate student at the time, so I appreciate that. All right, the title of this talk is called Stimulus Control, but perhaps it should have been titled Stimulus Out of Control. And one of the motivations behind this talk is I hope to inform you a little bit more about the work that we've been doing, but also to provoke you a little bit more and empower you with some tools to do more with your stimuli, because I started keeping track of the stimuli yesterday at the conference, and they're boring. They're still lights, they're still flashes, they're still tones. The world is far richer than that, and I hope to convince you of that today, and so on. But let me start off first with a real simple example of what we mean by stimulus control. This is the G-rated version, and it gets better as we move along. So this is what we mean by stimulus control. What we'll do is typically, and all these experiments will involve pigeons. We'll have two key lights. One will be red, one will be green, and even though we have them on computer screens now, they're still the size of Grayson-Stadler inline projectors. I don't know quite why. One will be followed by food, and one will not be followed by food. And we all know what will happen after enough pairings of this is the birds will begin pecking at high rates to this, and they'll peck at low rates to that. That difference in response rate, as Peter pointed out, is differential behavior in the presence of these two stimuli we call stimulus control. The animal's making a discrimination that's under control of stimuli and doing two different things at that time, and that's great. Here's a really good definition of it. It's the search, recognition, and discrimination of invariant structures among stimuli that discriminate and predict consequences. And in some ways, the invariant structures is the interesting part of today's talk. And it sort of picks up on that very nebulous phrase that was in the Thorndike's quote from the last talk, the situation, which is a very loaded term as we find out. But let's talk about a perfect world here with our discrimination where we have red and green. We're going to divide up each one of these stimuli into two properties. Let's call them invariant parts and variable parts. The variable parts, those are the fact that from day to day, the pigeon may be developing cataracts, and so it changes. At one time, it's standing over a side of the box, and the circular key looks elliptical. The house light's flickering one day, it's not looking quite as green as it did the other. All these things we need to learn to ignore because they're not predictive of reinforcement. What we end up having to do is talk about the invariant structure, which we hope is the essence of redness. And the same thing over here with green. We want to only get the greenness and ignore all those other parts of the stimulus that don't have anything to do with predicting what's going to happen. And because this is the Society for the Quantitative Analysis of Behavior, I thought it was very important for me to at least put something in here that had a mathematical part of it. There's an equality sign in there. Okay? Because where the land where we're going, we're not yet ready for mathematical descriptions of what's going on. Hopefully we will be, but at the moment we're not. Now, in the origins of the history of stimulus control, we got off to a great start. I think the first experiment in animal learning cognition really is the whole evaluation of clever Hans. And, he was going around doing the American Idol thing in Europe for a long time, impressing people. And it wasn't until a psychologist suggested to his grad students that they bring them into the lab and they start systematically decomposing the stimulus into different parts and figuring out what was really controlling the animal's behavior. Instead of these uncontrolled field observation, we bring them into the lab, we break it down, we find out the intelligence is in the observer and not the horse. And so it's great. And that stimulus analytic technique is going to still work with us today. But then we quickly got going, and Thorndike, and I figure the tech people at Columbia have gotten better at constructing things since then. Here, this is the situation he was talking about, and Curler with his work with the 3 boxes. Lots of interesting behavior going on, but the stimuli are really hard to quantify and understand. And when faced with complexity, what do all good scientists do? We retreat. No, we don't. We retreat into simplicity. All right? And that's what we've done now for about, 80 years, is we found ways to simplify the situation based on Pavlov's work, and then subsequently here the classic work of Gutman and Kalish, where we're going to train them up with a single colored stimulus, and we'll produce a testing series with a variety of different colors, and we get back the classic stimulus generalization for a beautiful example of stimulus control, something we can stop and appreciate. It's truly beautiful. The problem is this elemental approach. It's been very tractable, analytical, and it's been very productive. Lots of great stuff has been done using this particular thing, but I think it's perhaps too clean. All right? And what we've done by ignoring the rest of the stimulus is we've ignored a vast portion of the information that the animal could have potentially used in learning a situation. So this is my attempt at reality. Oops. No, maybe not. Oops. I'm determined to show this, so I'm going to go. It worked every time yesterday. Why is my movie not playing? There we go. Okay, now stop. Could be so exhilarating. All right, so reality. And what is reality? Reality is a sequential stream of invariant structures at a wide variety of different levels of description. Now, what do I mean by that? Well, what we saw in that short clip was a boat that went down the river. It then moved to an interior shot of the boat where we saw Hepburn take control of the rudder. Bogey goes to the front, fixes something, cavalierly takes out a cigarette, starts to smoke. They have a conversation. There are lots of events that are happening. Now, of course, I also saw a square brown box slide across the screen. I saw two sort of orange-colored blobs with various things on them move together and make some sounds. And I also saw a whole variety of pixels shift systematically from moment to moment to moment. Now, I think there are a variety of different levels to description of any one scene. And what we have done in this, in our science is we've hung out down here. And we have ignored lots of what's really interesting. So up here is the stream of reality. It's the causal texture of the universe that slides by us as we move through the field of attractors. And of course, it moves through time. And it's extremely complex. But we can begin to break down some parts of it. We can break down at least some parts that occur over time. We'll make them events. Events always take place over time. Some events that happened in that screen shot was the taking out of the cigarette, moving from the tiller, and so on. And any one of those events can be discriminated from another set of events. Behaviors, all of those kinds of things. It's a very loaded level. Of course, these are then composed of individual scenes. These scenes transform over time, but we can pull one particular scene out. Here's another picture of Utah, for those of you who were here yesterday. These scenes, of course, can be broken down because scenes are composed of multiple objects. And those objects exist as constructed from configurations, combinations of dimensions, which eventually all come down to the features that we register. All right? And here's another way of sliding through that same level of description. There are features. We'll just call those things that have perceptual existence. When we begin to group them along a continuum, we call them dimensions. We can then take some of those dimensional things, and when we get really wild and crazy, we make compounds out of them. All right, and so configurations are always multidimensional. We can also call them patterns. I use the word configuration compound interchangeably in this talk. What we almost never do is talk about objects. Objects look very much like compounds, but they are a little more, they're a little different from that, and they tend to be bounded. They tend to be three-dimensional. They have multiple viewpoints, and so on. Objects come together in scenes. Scenes I think of as just multiple objects in some kind of spatial context. And then events, which are always extended in time. They contain motion. They're dynamic. Behaviors are events. And they're also multimodal as well. Now, within any one of these levels, I want to argue, is there's a set of invariant structures and a set of variable structures at the event level, the object level, the configuration level, and the feature level all the time. And they're present all the time. All right? And what animals can do is pick any one of these levels, as human beings could do, and try to pick out the invariants and ignore the variable parts of those. And at some point, we may add a second stimulus where they're going to have different invariant and variable parts. And that's going to allow us to look at relationships between those two stimuli, such as identity, sameness, difference, logical relations, spatial relations, temporal relations, social relations, and so on. And these also turn out to have invariant and variable parts as well. And while I have some research looking at this particular question, we're going to really focus more on this particular aspect of this description today. And I think there's a certain sort of fractal quality to these levels as you go through them. If you think about the question of categorization, object recognition, categorical perception, they're really all the same issue, the same problem. They're just at different levels of description. And that level, question is, of course, how do you identify a variable stimulus that's a member of a larger group? So some relevant issues in stimulus control promoted by this view are that, well, can complex properties of stimuli be discriminated? It's pretty clear that dimensions and compounds those kinds of things. Configurations can be processed, but do animals have an appreciation for objects, scenes, and events? Not a question that's been well explored. We also want to understand what functional level of description occurs in any particular discrimination. I'm going to give you some tools for looking at that today. We also want to know what invariants are available at each level. What kinds of things are animals sensitive to at any one of these? What factors influence the level that's employed? I'm going to argue that pigeons and other animals can slide between these levels, and that there are tools that we can use to convince them to move to different levels. And another question is what common or unique mechanisms are involved in each level? This one's going to turn out to be really critical for us because we have very powerful tools and very good theories for understanding elemental and featural and compound discriminations. Now, are those going to scale up if we start moving to more complex levels of description, such as objects and scenes and events? If they do, great, we've saved ourselves a lot of time. If there are emergent properties at each of those new levels, well, then we have some work still ahead of us. And then, of course, we always want to conduct comparative analyses across species because they may vary. So what I'm going to do next is just give you a quick tour of different levels of control, and while we're doing this, hopefully give you a few tips as to how we can move forward in the analysis of complex stimuli. All these experiments are going to feature my animal, the pigeon. They're going to take place in operant chambers. They're all going to involve complex stimuli presented on computer screens. All right. We're going to employ a number of different strategies while doing this work. We're going to primarily feature the stimulus analytic approach that was outlined with Clever Hans. What we're going to try and do is present complex stimuli, then break them down into their component parts, and see which ones we actually need to keep to understand what the animal is doing. Some of the tools of the trade in that, we're going to do discrimination training. When we train up a discrimination and the animal comes under stimulus control in this situation, we're going to say they have representations that are different. If we can get transfer between two stimuli, we'll typically make the inference that there's some kind of shared notion between the two that's mediating the transfer. We're also going to examine patterns of errors, a very rich place for understanding representations because they tell you something about the similarity and structure of the stimuli. An important pattern to observe in all of these is that I'm going to use large numbers of carefully controlled, highly variable synthetic stimuli. So we're not going to just take the world as it is, we're going to construct it. That's a very nice advantage of being able to do things with computers. And we're also going to employ considerable variation, which is going to be helpful for provoking the birds to use different kinds of invariant structure. So the first question, and then the rest of the talk is outlined in a series of questions about various research projects that we've done that help sort of promote the view that I outlined just a few minutes ago. The first one is, are different spatial scales involved? All right. And probably the experiment that we've done that is most compelling on this particular issue is one where we looked at hierarchical stimuli. Right. Hierarchical stimuli are compound stimuli, and they have two levels, a local level and a global level. And what we're going to do is to make relevant stimuli. In this case, the relevant stimuli are the T, N, X, H, or the T, N, X, and H. The animal's going to have to report which letter it sees. And here's a local stimulus. What happens here is you make an O, which is going to be our relevant carrier stimulus. And it's made out of local T's, local N's, X's, and H's. What we're going to do up here is use our relevant carrier, the O, to make a global T. Now, when you do these kinds of things with human beings, and I present you a stimulus like that, most of the time you're going to first see a T before you see the H's that make it up. All right, so we tend to see the forest before the trees, and we report the global level before the local level. And what we tried to do in this experiment is to, A, get pigeons to report back to us both about the large-scale information and the local scale information. And we ran some controls as well. And what I'm going to show you first is some acquisition data. Oh, no, I'm not. I'm going to show you the procedure. And what we're going to do here is there are 4 keys. These keys are just the report keys. One is the T, one is the X, one is the N, one is the X. And we're going to represent the letter at either the local level or the global level in the center of it. Some things that we did that are, I think, particularly helpful in persuading the animals to learn the overall configuration is, A, the stimulus moves. It doesn't move within a presentation, but moves between presentations. So you can never predict where it is. So you can't learn to just stare at one spot on the screen and figure out what to do based on that spot. The other thing we did was we varied the size of it so you could never predict whether it was going to be the medium version, the large version, or the small version, which encourages the animal to look at the entire image. And what we found out, there are 2 things that are important in this curve. The first is that the local characteristics, the local relevance, this is the critical one here. They learned that one the fastest. The global relevant one was much harder for the birds to get together. Right? So there seemed to be this local dominance over the global characteristics as expressed in acquisition. Eventually, you can get them to learn both so that they're concurrently able to report to you the global spatial scale or the local spatial scale in the same experiment at the same time. All right. Now, an obvious question that comes up at this point is, well, is the information the same at both levels? All right, great, you taught them 2 discriminations, they're concurrent, but are they really doing what you wanted them to do, which was report back the letters? All right, and hopefully they are doing the same thing at each of these levels and changing the scale at which they examine the stimulus. And here's the data that demonstrate that. I'm not going to leave it up there very long for you to look at. I'm going to say, look, there are common mistakes that occur at each level. The X and the N are confused whether they're at the local level or at the global level. Okay? And so those common mistakes, that pattern there convinces us that, in fact, the animals are doing the same thing at each level. Well, so the next question we wanted to ask in a more controlled way was, is one scale more dominant than the other? You know, humans, as I said earlier, tend to be globally dominant. They report the global characteristics before they look at the local characteristics. So they tend to zoom in on the stimulus and break it down. Is the same thing true of pigeons? And one way to do this is to conduct a transfer test with some conflict pro-stimuli. In this case, what we're going to do is we're going to present a stimulus that's relevant at both characteristics. So, excuse me, you could report the global X, or you could report the local T. So what we'll do is we'll present these stimuli for a brief period of time, and then just simply let the animal tell us what it saw. All right? And we won't reinforce them. These are all going to be done in extinction. And here's the answer to the question. This is presentation time down here. This is the percent of the level chosen. You can't really talk about accuracy because there isn't a correct answer. But what we find is at very short presentation intervals, the animals tend to report back the local characteristic first before they report back the global characteristic. And with time, the global characteristic comes up. All right. So the local level seems to be more critical, at least in this experiment. So here's some conclusions that I draw from this particular experiment. Both spatial scales can control behavior, that the animal can be paying attention to things at a larger spatial scale and a local spatial scale at the same time. They can concurrently control behavior. Similar information guides both levels, and local information dominates global information, at least when derived from grouping. Is that always the case? Is that always true in all situations? I'm not ready to claim that. Pigeons may actually have some problems specifically with grouping separate and disparate information into larger shapes, which may be part of the reason that we get the results that we do. But at least in this experiment, local information tends to dominate global information, and that's different than what you find with humans. And then finally, if I were going to go back to my original levels of description, I'd say the stimulus control here that we've demonstrated is just simply at the configure level. There's nothing sophisticated about what's going on. The animals are simply matching patterns. They can do it at different spatial scales. That's great. I think that's consistent with the kinds of things that I was promoting earlier, but I don't think there's any objects or anything involved here. This is just simply pattern matching. Well, what about some of these larger scale structures I was talking about, objects and scenes. Do they exist for pigeons? One obvious question is, are pictures examples of control by objects and scenes? Because we've certainly, Herrnstein has started a whole cottage industry of people showing pictures to pigeons of all kinds of things. And do they really see the content in these pictures? Or do they see them simply as patterns? It's an old question and one that we've been exploring, partly motivated by the exemplar work that Tom mentioned in the introduction. One of the things that we've been doing with both pigeons and baboons is trying to see how many pictures or how many images they can remember, because it would be nice to know just how many associations they actually can learn. And so what we do is we typically present a picture that's randomly pulled off the internet of all kinds. And what we do in this situation is we randomly take each picture, we assign it to either be a right response or a left response. That's arbitrary, so all you can do is memorize each picture. We present them in blocks of 30, and we do this for years. Linus, my most experienced bird, has now been working on this task for 6.5 years, which is a good reason for working with pigeons. What we do is we give them a block of 30, takes about 16 days to learn that. Then we put that in the memory testing pool. We give another block of 30, they learn that. Then that goes into the memory testing pool, another block of 30. And we just keep adding 30, 30, 30. And then on half the trials, we test the old items, and the other half, they go to school and they learn some new items. And we just keep building up their repertoire of images and see what happens to performance. And this is what happens. So this is the number of items in the memory set. And there are two curves here. We'll talk briefly about the baboon. This is work I did with Joël Fagot. He did the work with the baboons. Baboons are prodigious memorizers of stimuli. In fact, Joël said we have to stop the experiment because after 2.5 years, they had remembered 6,000 items. And they were still at like 85%. And so all our estimates suggested that we were going to have to go out to 16,000 items before we might even get them down to chance. And he didn't want to do that. So I can appreciate that. Baboons, but pigeons, you can press them. They've got smaller brains, and so you can fill them up a little bit faster. Their performance begins to systematically decline as you increase the number of items. And we can do some mathematical modeling of this. And when we do that, we get an estimate that they're remembering about 1,000 items, even though we have more in the memory pool. At any one time, they seem to know about 1,000 of them and what to do when going right and left. Interesting fact, one of the things that we've wanted to do since then is, well, what do they know about these 1,000 items? Okay? Because it's one thing to know them, it's another to try and understand how they're all represented and stored and what they know about each one. So what we've been doing is a lot of various kinds of stimulus analytic tests to try and figure out what they know. So we test them with grayscale stimuli, we test them with stimuli that are blurred, we flip them and rotated them. We've done a lot of things. I'm only going to present a couple of them because I think they're informative for what I want to talk about. And the first one is, let's break up the overall organization of the stimulus. All right. So we'll take this stimulus, and what we can do is we can scramble it into 60 pixel blocks. All right, which breaks up the global organization and also any objectness that it may have. But it keeps some of the local features available to you. And one of the things that we're interested in this experiment is what's the sort of the spatial brain that they can latch onto to memorize any particular picture. Do they need the whole picture, or do they just need some spatial some feature of about that size, or at the 30 pixel size, or eventually the 5 pixel size. Okay. So what we're going to do is we'll take a bunch of these pictures, we'll put them in, and they'll either be in a complete whole or scrambled at the 60, 30, and 5. And let's see how fast they can acquire them. We know it takes about 2 weeks for them to memorize a set of these things. And so here's the learning curves for the two birds. And here's the whole picture right here. And right on top of it, and not different for either bird, is the 60 pixel curve. Right? So seeing the whole picture having some kind of coherent meaning for us doesn't seem to be at all critical for the birds. All right, scrambling it up, it's just an image, but it's not a thing. You do slow them down when you make the scale of the features too small, so there does seem to be a preferred spatial grain that they would like to latch onto. However they're memorizing them, they want a certain hunk of it to be of a certain size. But even when you scramble them down to 5 pixels, they can eventually get those just as easily. So the fact that the whole equals the 60, all of them are learned, suggests that picture content isn't important in this any way, shape, or form. That 60 is greater than 35 suggests that the size of the local features is critical, and that the 5 pixels learned at all suggests that global pattern memorization, just the look and feel of the stimuli, can be learned. Another kind of test we can do is we can re-scramble these things. So what we can do is we can take the very same image and then, like that game where you move the squares around, we'll do the same thing and just move these kinds of things. Will they notice anything different at all between these two? Impossible down here, I assert, although those are different. Up here, you might be able to realize that, that the head here, well, the head's actually in the same place, but some of these features have moved, okay? Will they notice? And the answer is, they do notice. Here's the baseline, here's the 5, which probably isn't really different for either bird overall, but at the 60 level, they do show a reduction. When you re-scramble the image, they say that's not the same image that I just saw. All right. So that suggests that they know something about the local features, and they also know where they go as a global pattern as well. So the fact that the 60 pixels reduced relative to baseline, I interpret that as the global position of the local features is encoded, that the 5 pixels equal to the baseline, it's just global pattern recognition only. There are no specific local details that are encoded, I don't think. I think they're sort of just memorizing the overall look and feel of that. So from this work, I don't think there's any evidence that picture content influences learning in this procedure at all. The spatial grain of the local features is important, although there's simultaneous control by local detail and global organization as well. They're paying attention to a lot of it. And we've even done experiments. What we do is randomly delete different parts on every trial so they never see the same image twice, and a different part of it is deleted, and they always learn those easily as well. I think what they're doing is basically memorizing the entire thing. Exemplar memory is really large for complex static information. And finally, I'd say, hey, stimulus control here is at the level of configuration of the pattern. There's no evidence that these are objects or scenes at all to the birds. Well, if that's true, then it raises this interesting question. Are pictures ever representational to pigeons? I mean, all this category work, is it something other than what we've thought? Well, I think the answer is, well, maybe it means what it means, but you got to do the right test to figure it out, all right? So one of the things that we've been doing is trying to, let's go back to this thing. I don't have to explain what this scene is to you, all right? All of you see basically the same thing, which are 3 objects in depth on a textured plane going away from you. The red sphere is in front, the yellow cube is behind that, and the red thing is yet behind that. And we all have the same perception of that because there are 3 different monocular cues here that help you decide that. First, there's this textured grain. Second, there's occlusion among the various objects to tell you which one is in front. And there's also relative size. Things further away look smaller than things that are close up. And so what we wanted to do was to see whether pigeons could understand the monocular cues to depth in this. And if they could, I would argue that suggests that they're seeing the same kind of scene here that we are. And the way that we do that is, I think you have to draw the attention of the birds to the right invariants. If you just give them a pattern, they'll just memorize it. They're really good at it. But if you arrange the contingencies of reinforcement in the right way, you can get them to attend to the invariant structures that you want them to. All right? And the key here is to ask them questions that where the represented content is critical. All right? So in this case, what we're going to do is we're going to say, can you tell us the depth ordering of these objects? And so the S plus is going to be this particular ordering. And there are 5 other possible orders of these very same stimuli. Those are all going to be S minuses. All right, so what you have to do is to find out when the stimulus looks like it's in this particular configuration versus any of these 5 other possible orderings. And then what we're going to do is we're going to systematically vary the various monocular cues to depth in the picture. So in the very bottom, we'll have the relative density, or the textured plane present. We'll have relative size, and we'll have occlusion. And very nice, salient, three-dimensional picture. Here we're going to just take, this is the great thing about using computers, is you can systematically eliminate one of them, occlusion. Here you can get rid of relative size, and here you can get rid of density. So we'll keep 2 of the 3 cues. Here we'll just let one of them be here, and then here we'll have the control where none of them are present. All right? And we're going to ask how well you learn each of these discriminations when your attention is directed to questions about depth ordering. All right? And the results are right here. The birds are fastest at learning the ones where all 3 monocular cues are present. All right. They're quickest when 2, or their next quickest when 2 are present. They're slowest when only 1 is present. And then, oops, they can actually even learn it when none of the cues are present. Okay? And the problem there is you can never underestimate birds. And it turns out that there's, when you light these things, there's also differential lighting on the surfaces of these things. This was a monocular cue that we had not appreciated when we did the experiment until after they started learning the control condition. But once we figured out that indeed that's what they were also paying attention to, and we uniformly diffused the lighting in that particular situation, then they can't learn it any longer. So there are at least 4 monocular cues to depth here, one we hadn't anticipated. But in all of them, all these results would suggest the pigeons are indeed sensitive to the monocular cues, and they're certainly consistent with the idea that they saw objects and they saw a scene here as well. All right. So I think this is consistent with the idea that there are scene-based levels of stimulus control, and at least under some circumstances, pictures can be interpreted as they were intended by the experimenter. Well, that's all well and good, Bob, but what's the key to all this? Well, here's what I think are the keys to generating scene representation in pigeons. First of all, you need very strong visual support for a 3D interpretation. All right? You have to have a good, high-quality picture with lots of support for that interpretation. Then you need to bring their attention explicitly to the invariant structures. In this case, the depth cues, and also the fact that you should pay attention to the global scale of everything, that are relevant with a representational view, and you do that by the contingencies of reinforcement. And what we do in our picture memory task is we simply ask, memorize these pictures. We don't tell them that the content of them is in any way important, so they don't bother to pay any attention to the content. But when we arrange the contingencies such that content should be critical, they do indeed get what we want. Another factor that's critical here is we present these stimuli at a distance from the pigeon. Many of our experiments, what we do is the pigeon is right up to the stimulus and they peck right at it. And so it's very much in their face. And we move it back about 14 centimeters. And I think that also helps them considerably in seeing the global overall relationships among the stimuli. You know, it's very hard to see anything when it's like that, even for us. And then finally, oops, let's go back here. And then finally, we present a large number of locally variable views. When we finally constructed all the stimuli in the experiment that I just showed you, we ended up having 800 different stimuli. Objects appeared in different locations. They varied in size. They varied in distance. We varied all the other properties that they could latch onto, leaving the only invariant structure, the depth relationships among the objects, as at least the most salient factor. And if you don't do those kinds of things where you're using large numbers of stimuli, I think they'll tend to slide down and give you a simpler result. Well, let's talk then about the final level. All right. Here I think my evidence is less convincing because it's a much more difficult thing, but do pigeons ever experience things like events? Okay. Now, for those of you, used to looking at colored lights and so on, since we're having to study events, things are going to have to start moving. Okay, so if you experience some queasiness, just close your eyes. The feelings will pass. So these are the accumulation of a number of experiments where we're going to ask about how pigeons perceive motion. And the first one, we're just going to simply ask, can motion be processed from an object-based discrimination? And what we're going to do here is we're going to have computer-animated videos where the pigeon is essentially going to see the camera, and it's either going to go through the object or it's going to go around the object. I'll show you an example of each. This is the through condition. Here's an example of the around condition. Now, the first 50 of those 60 frames are exactly the same. Only the last 10 frames differ in how the camera goes around. Well, can the pigeons learn this kind of discrimination? What we do is present this in a go, no-go situation where we present the video for 2 seconds, and then we repeat the video, so the entire thing is on for 30 seconds, and they see repeated examples of the same video in a row. And we define one of them as the S+, let's say the around, and the negative is defined as the through, and what we do is look for peck rates to repeated presentations of these particular videos. Takes them a couple of weeks to learn. We can then transfer them to new objects to see if, in fact, they're really picking up on the motion part of it, or whether they're just picking up on some kind of simpler two-dimensional cue or some other simpler transformation of the object. So we present a variety of different objects. All of them they transfer to. We can eventually combine them all together, which is another example of multiple exemplars helping pigeons to identify complex invariants. Which leads us then to another interesting question. Is motion continuously perceived in these videos? All right, and how would we ever be able to find out the answer to this question? All right, and the answer is it is continuously perceived, and it comes from doing analyses like this. So what we're going to do here is this is the stimulus interval. So the stimulus comes on here, goes off here after 25 seconds. So within that 25 seconds, each video is played a number of times. Okay? And what you see here is the onset of the video, pigeon peck rates first come up. All right. And then as soon as the event happens, they go down, but the two things begin to separate, the S+ and the S-. Then there's this long run-up where the stimulus is uninformative, right, because that long run-up to the event is the same in both ones. So there's a little bit of loss of stimulus control, but then the event happens and positive peck rates go down, negative peck rates go down, and each one of these lines that you can barely see is a repetition of the stimulus. But what you see is this regular cyclical observation of the pigeon as it sort of gains stimulus control when the event happens and then loses it momentarily when the events look similar between the two videos. And you can see that both for the coherent one and for the short coherent one, and then for the long coherent one. So some videos are longer, and that strings out their behavior in a longer period of time, suggesting that they are watching these videos the entire time they're on and doing what's appropriate based on what they're seeing. Is coherent motion critical to these discriminations? And what because events always occur in an ordered, sequenced fashion. If you scrambled them, they wouldn't necessarily be events, although see Memento is an example of a movie where that's not true. But we wanted to ask whether pigeons needed coherency to see these kinds of events. And so what we did is we had two groups of pigeons. One group of pigeons saw the movies presented just as they should be, in a nice ordered sequence. The run-up, the event happens. The other saw the exact same set of videos, but we randomly scrambled the frames that they saw, so that it would jump to the end and back to the beginning and to the middle and so on. And we were going to ask, would that disrupt their perception of these video sequences? And the answer is twofold. I'll point to the one problematic one. Here are the two random groups that we ran. Both of them learn that. So even when you randomly scramble these things, they can still get information about whether it's an around or through event. Why is that? Well, okay, we all figured it out. There are some frames that are really diagnostic. We could stop the video where late in the game, where the video and the object is off to the side versus symmetrically around you. That's a simple 2D spatial cue. And if you didn't have to have the motion to see that, you'd be able to know whether you were in the around or through condition. I think that's what these pigeons can do here, is they can pick up on that frame no matter where it happens and do the appropriate thing. So there are some 2D cues that don't have anything to do with motion or events that happen here. But we do find a coherent superiority effect, and that's the difference between this group here and this group. This difference I interpret as the fact that the pigeons do really need coherency to see the motion component of this. So it happens to be that these particular stimuli, there are some 2D cues we didn't control very well, some 3D cues based on probably motion that are important, and when we have them, that helps. Which motivated us to, can we get rid of these 2D cues? And there is a way to do that, and the way you do that is you, do these move? Yeah. Is you can just rotate them, and you can either have them rotate right or left, and then it's essentially the same video backwards or forward, and the only way you can make out which way it rotates is if you integrate over time, because if you stop it at any one point, you don't know which one is the right word and left word, rotating one, until I make it move. That gets rid of that little annoying 2D cue problem. Birds easily learn this. Again, it takes a couple of weeks. And then when you randomly scramble everything, it goes away. So when you devise a discrimination where the 2D cues aren't present, they do and can pick up on the motion and maybe the events that occur in these particular videos. Finally, can they tell us anything about the motion itself? Can they tell us about the properties of motions that occur within these videos, independent of the objects? And we were trying to think of a way to do this, and what we did was to ask them, could they tell us the difference between fast events and slow events? Okay. And so what we wanted to do is to make sure that they don't pay attention to anything having to do with transformations of particular images and so on. We generated lots of different videos of objects rotating around their axis. And the discrimination now is, if the video moves quickly, you see the thing spin quickly, that's going to be the S+. If it moves slowly, that's going to be the S-. So we're just simply going to ask them to tell us something about the property of the event, independent of what object is producing it. All right. Turns out this is also a really easy discrimination for pigeons to learn. Within a couple of days, they're showing you discrimination between the S+ condition, the fast condition, and our S- condition. And within a week, they know it very, very well. So telling us something about the speed of occurrence that an object is transforming at is something they're sensitive to. And we tried some transfer tests. Transfer tests consist of new objects that they haven't seen before. They passed that just fine. Rotating in another axis that we trained in fact, the two other axes that we didn't train in, they continued to discriminate fast from slow just fine there. And we did new motion translations, so we had the object not rotating any longer, but what it did was it moved around the screen either in a fast way or a slow way. We tried 2 or 3 different translation paths. Those they could all continue to tell us whether something was fast or something was slow. When we began to change the color of the object, because this is something you can also do, it doesn't happen in the real world, but you can slowly, in these kinds of computer worlds, change an object from being red to green. And that can either happen quickly or can happen slowly. We can also do the same thing with size. When you do those kinds of things, we don't see any transfer. So it seems that whatever they learned about fast and slow was not about change in the world per se, but particularly the path or the motion of the object itself, and not a property of the object. So the conclusions from this last set of experiments are that there are continuous perceptions of video. They can perceive things dynamically and integrate them over time, which is going to be critical. Smooth, coherent motion improves object-based motion discriminations, and both object motion and motion relative to objects can be discriminated. Now, is this stimulus control at the level of the object and the event? Well, okay, I won't put scare quotes around them, but I'll put a question mark at the end. I'm not sure that they're the best evidence we have, let's say that they might be able to do the kinds of things that I suggested earlier on. So the conclusions that I want to try and bring out of all these different examples is that animals can recognize invariance at a variety of different levels of description. We have been fixated at a very simple level of description, features, dimensions, and compounds, but I think it's with the right kinds of techniques you can get evidence that they can also pay attention to invariant structures and be under stimulus control at the level of the object, the scene, and even the event. Animals may tend to employ the simplest level that's available to them, and so you have to guard against that carefully. One way to do that is to use multiple and variable exemplars. What this allows animals to do is to identify more complex invariants in the stimulus than they otherwise could. That if you ask them to do something with simple stimuli, they'll do something simple. If you ask them to do something complex and you ask them in the right way, they can do that as well. The organization of stimuli and task demands is critically affects the invariants that are extracted, and I will argue that these higher levels of stimulus complexity have been systematically under-examined, and we need to go about changing that. Future questions and directions. I guess, can dynamic events be sources of stimulus control for animals? I think they can be. I think they have to be. And I think what we need to do is to find ways to begin to ask those kinds of questions in a systematic way. The people in the human cognitive literature are certainly beginning to explore that, and we need to find ways to do that as well. We also need to understand better how time, space, and events are incorporated into representations, that the kind of simple little associations and image-based exemplar memories that we have now just aren't going to be up to the task of really representing how reality is stored, and understanding event and perception and event memory is going to be critical. Are there basic levels of descriptions? I don't know. Roche has proposed that certain basic levels to objects, and that the subordinate and superordinate level of objects are not as natural. The same thing may be true of events or scenes. I think those are some interesting questions to explore. And whether there's change across situations. And then finally, something that's really sort of critical for the whole enterprise that we've built here is, are there emergent properties in the nature of the mechanisms that are used at different levels? We have great understanding of stimulus control with fairly simple stimuli. And if all our tools scale up, it'll be grand. But if it turns out that there are new properties in the stimulus and in understanding events, then we're going to have, like I said, more work ahead of us. So these are the various people who have collaborated with me over the years. I appreciate all their help and also the support of the National Science Foundation in this work. And I appreciate you letting me put you under stimulus control for the last 50 minutes. Thank you very much. Yes?
You showed early on, I think you can retain thousand images and that room to go on several more thousand.
Yes.
How many, I would say,
well, the question is, would humans show the same patterns as the baboons and the pigeons if you could get them to do this? I did try to convince a couple of undergraduates to do this every day of their senior year, and they did it for 2.5 weeks and then abandoned the project. I don't know. People have studied image memorization in humans using recognition procedures where you present one image, for 10,000 of them, and then you come back with this one-shot presentation. And we're very good at remembering those kinds of images. But what the pigeons and the baboons have to do is they also have to remember an association. They have to remember not only the image, but whether it's a right or a left. And I don't know if humans would be as good at that as the animals. It's a good question. I'm going to have to pay somebody to find the answer to that out. Because getting them to volunteer for credit won't work.
But you're in the home of the enforcement. Everybody here could have told you you're going to have to pay it.
It's contingencies. It's all contingencies. Yes?
It's been suggested that pigeons won't show good identity matching readily with pigeons because they respond to the entire configuration of stimuli on the display as opposed to learning specific if-then conditional relations. Would you conceptualize that as sort of the global-local distinction that you've been making here? And number one, and number two, if yes, what you talked about ways to enhance control by the global dimensions. Are there strategies to enhance control by the local dimensions to reduce identity matching in pigeons with status?
Well, let me answer the first question. The first question was, there's some evidence that the pigeons have difficulty learning identity matching in matching sample tasks because they end up memorizing the configuration. And I think they often can memorize the configuration. And probably what we should do, and we don't do it, and I guess it's just because we build our devices after mechanical devices. Why do the test stimuli always have to be on the side, and why do they always have to be static in these kinds of situations? If you want them to learn the relationships between things, then the test stimuli and the samples should move all over the place all the time. I think if you did that kind of thing, the pigeons would stop trying to memorize the global configuration of these kinds of things, I think. And also having thousands of different examples really helps too.
Well, isn't it true? I'm not sure if this audience all knows that the question of whether pigeons can do the same discrimination has been solved, and the answer is they can. When you use lots of stimuli, lots and lots of stimuli, they can solve the same.
Yeah.
Okay.